All right, my name is Jonathan, and I'm doing my presentation on citrus screening. This is my abstract. I'm not going to make you guys read all of that, but basically what I'm saying is there is a disease called Hong Wang Long Bing. It originates in Asia. I'm going to call it HLB. It comes from a bacteria. It's spread through a specific insect. It's called an Asian citrus psyllid that I'll be calling ACP for short, and it originated in Asia in the late 1800s. Recently, it has migrated over to Florida, and it has been affecting our citrus, which makes it a very dangerous and possibly very harmful to the citrus industry in Florida. More on the disease. Like I said, it originated in Asia in the 1800s. There's been cases in those places. And in 2005 is when it first came to Florida. And most infected trees die within a few years, but even beyond that, just the psyllid itself harms trees because it eats the new growth leaves within the orange tree and inhibits the tree from growing, and eventually could kill it that way as well. It is caused by that bacteria, and like I said, it's transmitted through this little bug here. It's about the size of a pin of a needle, and it damages the citrus by feeding on the new leaf growth. It lays its eggs on the growth, and it transmits the disease whether it gets from eating a tree that's been infected. And this is what the disease looks like after a tree gets infected. The leaves themselves experience a yellowing. They spread throughout the tree, and the fruit itself, which is the main problem, turns into a greenish color. Basically, it overripens while it's still not ready to be consumed, and the fruit itself gets it's not suitable for eating, for juicing, or anything like that. It basically becomes useless. More statistics on the disease itself is basically the citrus industry in Florida is worth $10.7 billion. And it supports 64,000 jobs. It's one of the more famous things in Florida that we're popular for. And recently, because of this disease, the USDA just allocated $31.5 million to research it. And as we've experienced because of it, 59% fewer oranges have been produced since the 90s. Partially from this and also for other things as well. And it's estimated that this disease, specifically in Florida, has caused $4.5 billion in crop damages and 8,000 jobs. All right, specifically about chemical engineers and what we are doing to fix this problem is through research, specifically with that grant that I mentioned earlier, that $30 million that USDA gave towards citrus screening. 13.5 of that has gone to UF, a lot of it to the agriculture department, and some of it obviously to the community department and others as well. My specific team is actually through the Particle Engineering Research Center, although all of the undergraduates working on it are chemical engineers. And what we are doing is using a dye-doped kaolin clay, which is a clay commonly used in makeup. And we are using FDA dyes, just blue, red, stuff that you would eat on a normal basis, like an M&M's, for example. We color the clay, and then it gets sprayed onto the leaves through an irrigation system like you would normally water them. And the idea is to prevent the ACP from landing on the leaf because it changes physically, and to prevent the insect itself from being able to see the leaf that it wants to eat because it gets confused because of the difference in colors. And this is what the dye looks like on the leaves after it's been coated. And as you can see the label here, we use a blue 1 dye. This would be ideal. It's pretty much fully covered on the leaf. As you can see, it's kind of hard to get all the leaves, as this one here is not colored, but that's also because you were targeting specific leaves when we were doing this. But as you can see, it's fully covered. The leaves is changed color, and there's a layer of clay on top. And that in itself prevents the bug itself from being able to eat the leaf because the bug is so small, it's hard for it to break through the clay layer. Specifically, what I was in charge of with my group is actually quantifying the color changes, because in general, color is a very qualitative subject. It's something that, this one looks like it's more blue than this one or yellow. But we needed to get numerical values so we could actually have hard data to say, this specific dye would work better towards preventing this disease. And What we chose to use for that was this, a spectrophotometer. It was a machine from Hunter Labs, and it works in this way. Light source is imaged onto a sample. A fraction of the light is reflected onto the sample. The light from the sample is then imaged through an entrance slit, and then the slit separates the wavelengths to basically just measure color. So basically, we all know like the Roy G. Biv scale. It just separates the wavelengths after it shines light on the sample. The light reflects back, and it separates the wavelengths to determine the most common color from the sample. This is what it looks like. It's really small, it's portable, and allows us to use it in the field too, which is one of its big advantages. The color itself is on two different scales. We use the tristimulus and the colorant strength. Both were, here's actually a little bit easy to explain. This one's here's color and strength. It's basically just, it has to do with the blueness, how red it is, and how light the color shines. And this one, the example is just the change from just basic leaves that we measured before we applied the dye and afterwards. And you can see here that applying all these dyes changed the most, with violet being by far the most change in blue. It added a lot more blue to the leaf, which is what you would expect. But that's good data. I mean, showing a change in 30 points values units is actually a change of about 50% in the color of the leaf. And this is the tristimulus scale, which is the more commonly used color scale. It's kind of like the RGB that we're used to, but except it completely gets rid of negative numbers by standardizing them just using some math and algebra. It's by far the most commonly used, and as you can see, there is still a lot of change, specifically in violet 2 as well. This so far has worked really well, and labs are actually working with Lake Albert in South Florida to apply it in the field on a larger scale to see how the psyllid likes it, or doesn't like it, I guess you could say. And as I said, we're experiencing good results with the psyllid not being able to bite into the leaf very well, not being able to actually find the leaf that it wants to eat. Moving forward, we want to see how the dye itself affects the photosynthesis of the leaf to make sure that oranges are still being produced at the rate that they need to be. And also if we can make the dye last longer. In other words, if we can apply surfactants or things like that that aren't harmful, that allow us to be able to recoat the dye less often, save money. Concluding statements, there's been a large surge of funding to prevent this, and there are plenty of research groups like mine that have various ideas and various solutions for this problem. And moving forward, we hope to be able to continue the great citrus industry in Florida. Does anybody have any questions? Yeah.
Did this have anything to do with the blue trees that were on campus last year?
It did not. The blue trees that were on campus were actually just paint. But it's a similar concept, though. We're just putting some sort of dye on a tree, and it changes the color, which confuses the insect.